Okay, good afternoon, everyone. I'm Hua-Sheng Chiu. I'm from Baylor College of Medicine. So it's really a pleasure to be here with you today. Today I'm going to talk about, there's a translation here, pretty cool. Today I'm going to talk about our latest research on lncRNA target predictions. So to begin with, let me first introduce the 6 compelling reasons why we want to study non-coding RNAs. The first one will be non-coding RNA can be they do not encode proteins, but they still can have regulatory functions. Second one, they can be transcribed almost virtually from anywhere from the genome. The third one is their size come from different sizes, because it could be as tiny as 20 base pair or as large as 5,000 base pairs. Because these 2 features, it's very difficult to predict non-coding RNA genes. The fourth one is our recent analysis suggests almost nearly a half of our transcriptome are non-coding RNAs. So this number is outnumbering their protein coding counterpart by a significant margin. The next one will be, in addition to their abundance, they are also very specialized. They can be tailored to each cell type, tissue, or even tumor type. The last one is, unlike typical regulator like transcription factor or RNA binding proteins, they can only regulate their targets across one regulatory modality. However, non-coding, they are often can regulate the same target across multiple modalities. So we will go deeper for this part later on. So in the past, we want to gain a better understanding about non-coding RNA tissue specificity. So we joined forces with an international consortium, as known as the RNA Atlas, in 2021, and we profiled almost 900 normal tissue samples. And we have successfully identified thousands of novel non-coding RNA species. In my lab, we apply our target prediction algorithm to predict targets for these novel non-coding RNA species, and eventually we assign their functions. So this work has been published. So now, in the past, my lab has been working on using machine learning or artificial intelligence to unlock the secrets of non-coding RNAs. We focus on microRNA, lncRNA, and circRNAs. We are trying to make all kinds of target prediction algorithms, including Hermes, qubit, long home, and big home. One thing I want to point out here is both long home and big home, they can make target predictions for lncRNAs. However, our findings suggest big home can have a much better performance than long home. So we will go deeper today as well. So these tools are currently available at our OpenRNA website. Yeah, we are happy to get this domain. This is not easy. And also these tools are pretty instrumental in understanding of the regulatory function of non-coding RNAs. So now, before we talk about big home, I want to introduce why we want to study, why we want to create a new algorithm. So,
sorry, we're sharing the wrong screen here. Sorry to interrupt.
No problem. Okay, no problem. Yeah. So before we talk about big home, let me first introduce the limitation of long home. So long home has 2 major components. The first one is called long home TR. As you can see, long home TR is used to predict transcriptional target of lncRNA and also the binding sites. So it assumes each lncRNA should have a direct binding on their target DNA promoters. And also they assume lncRNA can regulate their targets through modulating their effectors activity. Another component is called long home PTR. It used to be predicting the post-transcriptional targets for lncRNA. While long-form PTR, you assume indirect competition for effective binding between lncRNA and the layer targets. So both components, they require sequence evidence and the expression evidence. For the sequence evidence, long-form TR always assume lncRNA can form a triple helical structures on their target DNA promoters. However, for the long-form PTR, they assume the lncRNA and the layer target, they should share a significant large common effector program. So I'm not going to talk about in detail here. So now we want to compare the performance of long-form TR and PTR with triplex predictions. So we download Pentagon-6 antisense long-uncoated RNA knockdown data. We further separate, we only use the ASO knockdown data with good silencing efficiency, more than 50%. So in total, we got 155 ASO lncRNA knockdown data. We further separate this set into two subsets. One is for the cytoplasmic lncRNA. Another one is for the nuclear lncRNA. Based on Pentagon-6 lncRNA localization classification. As you can see from here, long-form PTR does have a theoretical advantage over other two methods because it was designed to predict post-transcriptional target, so it performed well in cytoplasmic lncRNA. However, if you look at a long-form TR, it doesn't have a theoretical advantage over other two methods. So it's obviously we can see long-form PTR can do well. However, we still have a great room to improve for the long-form TR. So that was the motivation. We want to develop a new algorithm called Big Home. Big Home is designed to improve the transcriptional targets and the binding site for lncRNAs. So as you can see, the cartoon here is very complex. So if you don't understand, please look for me after my talk. So in brief, each lncRNA under Big Home framework, it has a random lasso classifier. And each random lasso classifier includes sometimes thousands to millions lasso regression models. So you may know each regression model has independent variables and dependent variables. So we calculated correlation between lncRNA and the layer target using gene expression data we downloaded from TCGA across all the 33 TCGA tumor types. And we use the correlation profiles as a dependent variable. For independent variables, we use GAPI-K-mer. What is GAPI-K-mer? It's a K-mer with some certain length, but it allows some mismatches. So it means it doesn't require stringent sequence matches. We allow some mismatches. So it's kind of fuzzy manner. So we ask for each GAPI-K-mer, what's their frequency on their target DNA promoter? Eventually, you can construct the frequency matrix. We use this matrix as our independent variables. So random lasso adopts an iterative scheme. So it runs over and over until they can generate predictive GAPI-K-mer. So assuming we have a predictive GAPI-K-mer after running the random lasso, we further ask, do we have any pair of predictive GAPI-K-mer? Once we combine their frequency profiles, we can even make better predictions. So now that's the reason we build a synergy network. As you can see the network, each node represents the GAPI-K-mer. If you can see any interaction between them, it's called a synergistic relationship. It means these two GAPI-K-mer, they have some synergy between them. And then we identify strongly connected component within this network. Each strongly connected component, they will be associated with one position-weight matrix. We use a zero-order Markov model. And we use each PWM we derive from this synergy network to make the binding site predictions. And then you might want to ask, does Big Home really perform well? So we compare Big Home, long-home TR, and the triplex predictions. As you can see right here, Big Home predicted binding site are more enriched at regulatory hotspot, including of co-promoters and the ATAC open commuting regions. So here we use TF binding sites as our gold standard, as our positive control, because we believe their binding sites are relatively well studied. And also we have a random prediction. How do we generate random prediction? Essentially, we randomize while maintaining the dinucleotide frequency for the promoters. And then we are trying to identify triplet structure within this randomized promoter. So we use random prediction as our baseline label. And then we also knock down multiple lncRNA using CRISPR-I. And then we measure gene expression changes using RNA-seq. As you can see, the big cone is in red. It always have a much better in terms of F-score or feature exact test p-value. You can see in the x-axis is the number of targets predicted by each method after sorted by their method-specific scores. As you can see, no matter how many targets are included, big cone can, generally speaking, can perform much better than other methods. Now, I'm asking you to imagine if you can imagine lncRNAs are a boxer, right? So lncRNA can deliver one single punch to the target, whether through transcriptional or post-transcriptional regulation. However, because it can only regulate one modality, we believe this impact will be very limited. However, if lncRNA can deliver a powerful 1-2 combo to the same target, we hypothesize this impact on the target expression should be much stronger. So based on our analysis, we identify almost 100 of targets there. We also define this kind of target as coordinated targets. According to our analysis, we identify 100 of targets that are coordinated. So now we want to ask whether the coordinated target can really have a stronger association with their regulator lncRNAs. So we use the same CRISPR-I RNA-seq data. We do see coordinated target have a more profound differential expression than the exclusive targets. And the difference between coordinated target and the exclusive targets, the difference are also significant. Now we choose to focus on GFS1. We want to investigate GFS1. We knock down GFS1 using siRNA, and we profile gene expression changes in 3 cell lines. Then we combine 3 RNA-seq data to make this volcano plot. As you can see, DICE1, after knocking down GFS1, DICE1 is significantly downregulated. And we also predict, PICOM also predict DICE1 is a coordinated target of the GFS1. And also PICOM predicted targets are significantly enriched in the differential expressed genes, same as the long-term PDR predictions. And now we want to confirm whether DICE1 downregulation can be confirmed by other biochemical assays. So we further knock down GFS1 using siRNA, and we measure DICE1 downregulation, RNA and the protein downregulation using RT-PCR and Western in 11 cell lines across 7 tissues. As you can see here, DICE1 expression is generally consistently downregulated after knockdown GFS1 using RT-PCR. As you may still remember, we predict DICE1 is a coordinated target of GFS1. So I will expect the protein downregulation of DICE1 should be even stronger. As you can see here, the protein downregulation of DICE1 is more than 45% compared to their RNA downregulation, it's only 37%. So the numbers showing here are average across all the cell lines. Now, you probably understand, you probably know DICE1 is a very famous microRNA biogenesis regulator, right? So we want to know whether GFS1 can regulate microRNA biogenesis through DICE1 regulation. So we further knock down DICE1 and GFS1, we measure microRNA expression using narrow string microarray data. As you can see, we also profile in 2 different cell lines. As you can see, in the x-axis is after knocking down GFS1. In the y-axis is after knocking down DICE1. The layer microRNA expression is almost highly correlated. The p-values are already under machine precision. So we believe GFS1 can regulate microRNA biogenesis through DICE1 regulation. So to finish off, so we developed PICOM to predict, to improve transcriptional target predictions. We also suggested PICOM predicted binding site are more enriched in the regulatory hotspots, and they are also more enriched in differential expressed genes. We also got all kinds of biochemical assays, including phenotypic assays. Because of the time limit, I cannot talk about them. So in closing, I really want to appreciate our wonderful collaborators, including people from Baylor College of Medicine, people from MD Anderson Cancer Center, Tsinghua University, University of Ghent, and also National Yangming Jiaotong University. A special thanks goes to Ting Wen Chen and Dr. Chong Wei Zhang. Also, I really deeply grateful for our generous support from NCI, CIPRI, and also European EU. So I'm happy to take any questions. Thank you.
Okay, I have one question.
Sure.
You use the random lasso. It is my first time to hear about random lasso.
Yes.
Is it better than randomness in general?
So the random lasso is actually a combined term between random forest and the lasso regression. Everyone is aware of lasso has some disadvantages, such as you cannot distinguish if there are two highly correlated features, right? If you run random lasso, only one of them will be selected. But based on this random lasso scheme, both correlated features can be selected. That's one advantage. Another advantage is random lasso. They only use a subset of features, but so they don't need to worry about overfitting problem like a lasso regression could have. Yeah.
Okay, thank you very much.
Thank you so much.